Hello, everyone. I'm Andrea, and I'll be talking to you about the garden of forking shortest paths. That is about a question of how do we analyze our network neuroscience data. So you might wonder, well, what is network neuroscience? Some of you might not be network neuroscientists. And in a nutshell, the idea is that we want to go from this, that is a brain, to this, a network. Using something like this, so functional MRI, there are other ways of deriving a network from brain data. And you might wonder, well, but why would I want to get a network out of brain data? And in a sense, the idea is that fundamentally what the brain is a network of cells. So networks should be an appropriate representation of brains. Unfortunately, we cannot actually look at cell-level networks of the whole brain. However, we also know that brains are very complex. I'm not expecting anybody to be surprised by this. And therefore, we need a way to talk about brains that simplifies what we know about them. And we are lucky because the mathematical branch known as graph theory provides a way to have detailed formalism and detailed understanding of networks. The idea, therefore, is to take a brain, find a way to turn it into a network, and then derive insights about that network from graph theory, and hopefully translate those insights back into brain insights. And here shown as just some common measures that we can derive from a brain network. Now, here is the problem. And the problem is that these networks all look very different, and yet they are all derived from exactly the same brain data of exactly the same individual during exactly the same scan. So what is going on here? Why do we go from the same brain to a multitude of networks? And that is the garden of forking paths. That is, to go from a brain to a network, there are very many steps that one needs to take, and each of these steps can go in multiple directions, and as a result, we have a combinatorial explosion. You might say, well, but does this really matter? Maybe it doesn't, because maybe the results are all the same across different network derivations, so I don't particularly care which of those very many paths you took. And we were wondering, yes, but are these results all the same? We do not know. So let's find out. So in this study, we had 7 different data sets of pharmacological resting state fMRI. That is because we had access to those data sets, but also because that is what we actually work on. All of these data sets have been used or are being used for publications, including network neuroscience publications. So these are real case studies, if you will. And there are many different ways to get measures out of a network, out of a brain network. And here we focus on 8 whole brain graph theoretical measures, that is not including measures that only focus on particular regions. And I will explain why in a second. But part of the reason is that these, being whole brain, are comparable across different network parcellations. And finally, we had 180 unique pipelines to go from same brain to a particular network. And for each of them, what we were interested in was consistency across the various pipelines. So what we did is we compared what the results would be when thresholded at the standard statistical significance of p less than 0.05. And what we are especially interested in are cases when the same data set analyzed in different ways produces diametrically opposite significant effects. That is, in one case, you find a significant increase, and in another case, a different analysis of the same data in terms of the same measure gives you a significant decrease. Because that is the kind of question that we're especially worried about, because if that happens, we do not know what the truth is anymore. And so there are many steps, and I'm going to run you through the ones that we took here in terms of what directions do we go for analyzing our data. And the first step is, how do we define nodes? Because networks are made of nodes connected by edges. Nodes are the dots, edges are the links between them. So what are our nodes in the brain? As I said, we cannot use each individual cell as a node, so we have to find a way to group them together And all of this is in the context, as I mentioned, of resting state fMRI. So what we do is we have different ways of parcellating the brain. And on the top, you can see the Lausanne parcellation. That comes in multiple scales, and in this case, we use what is probably the most common of them, that is the scale with 234 regions of interest, that is ROIs. And that provides whole brain coverage. Here we excluded the cerebellum for reasons that I'm not going to go into right now. But the point is, on the left, you can see the actual parcellation, and then on the right, you can see a representative functional connectivity matrix derived from it. And then here we use instead the AAL parcellation, which is the most commonly used parcellation in network neuroscience, at least in the clinical network neuroscience literature. And as you can see, each region is more coarse-grained than the ones in the Lausanne parcellation. And here, once again, you can see a representative functional connectivity matrix from exactly the same subject as above. And finally, we also use the third parcellation that only comprises 30 ROIs, and that is quite different from the top two because it's derived from independent components analysis of about 400 human connectome subjects. So each of these ROIs belongs to one of the common resting state networks. And crucially, this does not provide full brain coverage, but only partial coverage of the cortex. Now, the next step that we need to take once we have defined our nodes, and this is a step that is independent on how we define our nodes, is, well, how do we define the edges? So the connections, again, between the nodes. And there are two different steps that we need to take within this decision point. One of them is, what time series do we use? That is, since we are talking about functional connectivity, we have one time series for each node, and we need to relate them in one sense. But first, we need to decide what do we use as the time series, because we could use the standard Bolt signal fluctuations as the time series, but some people also prefer to take the instantaneous amplitude of that by means of the Hilbert transform, which is supposed to perform data cleaning in some sense. And once we have decided on that, we have the next question, and that is, how do we relate those time series to each other? Now, the most common approach is to use Pearson correlation between them, relatively straightforward. One problem, though, is that if you're using Pearson correlation, you're going to have negative edges. That is, two time series might correlate negatively with each other. In fact, that is well known to happen for networks such as the default modes and the frontoparietal network. And that can be a problem because not all graph theoretical measures are able to deal with negative edges, and it is not clear in some cases what they would mean from a biological perspective. So a different option is to use the absolute value of the Pearson correlation. That is, just take your negatives and turn them into positives. Straightforward. And in the figure here, you can see what the results would be. So in blue is the same time series correlated just using Pearson correlation. And on the right, and in red, you can see what would happen if we took the absolute value of those results. Now, there is also a third option, and in fact, there are many more. The third one is just the third that we considered, and that is using a way of associating two time series that does not, by definition, provide negative results, but only 0 or positive. And in this case, we use normalized discrete mutual information between the time series. Now, once again, after we have made these choices, which are all independent of each other so far, we have to decide how to threshold our functional connectivity matrix into an adjacency matrix of a network. And the reason why we need to do this is because the idea is that brain connectivity is, in some sense, sparse. That is, not every single region is connected to every other single region. However, functional connectivity matrices, since they are derived from simple correlation or mutual information between the time series, are by definition dense. That is, there is going to be a value in each cell of this matrix which represents the connection, as we defined earlier, between time series 1 and time series n. So the question is, which of these do we exclude as spurious and which ones do we instead retain because we believe they reflect something that is true about our data and the underlying brain? And the problem is there are very many different ways of thresholding a functional connectivity matrix and deriving an adjacency matrix out of it. A most common option is to threshold by density. That is, we're going to say we're going to keep the top 10% or the top 20 or 25% of the edges in this matrix, and everything else is going to be set to 0. And this is known as proportional thresholding, because we're keeping for each network, for each matrix, the same proportion of its entries, and it can be more or less sparse. So, not sure what's going on there, but let's keep going. So in this case, this is probably the most typical approach, that is taking between 10 and about 25% of the top strongest edges, and then deriving results for each of them and averaging together. However, other people choose much denser networks, and so we try to reproduce that by doing the same thing between 30 and 50% of the edges in our networks. Another option that we came up with was, well, we also have, in some cases, structural connectivity data, either for the same subjects or, in this case, from about 1,000 people from the Human Connectome project. So what we did in this case is we took those DTI data, and that is how you get a structural connectivity network. And the advantage of DTI data is that the network that they produce are naturally already sparse. They don't need to be sparsified further in order to derive an adjacency matrix from that. So what we did is we took those DTI data and we parcellated them according to the same parcellations that we saw earlier to derive a particular structural connectivity network according to that parcellation. And then we said, okay, we're going to threshold our functional connectivity matrix to have the same density as this corresponding structural connectivity matrix. And as a result, the density will be different depending on which parcellation one used, but it is still a proportional kind of threshold. Now, an alternative is to use instead absolute thresholding. That is, proportional thresholding will impose that the same number of edges should be present in two networks, regardless of whose networks those are. But a problem with that is that some people, in some cases, for instance, as a result of some psychiatric condition, might have a larger or a smaller proportion of edges in their brain networks. And so we try to get to that by using absolute thresholding. And the question, of course, is where do we put the threshold? Well, we thought a natural way to put the threshold might be at the significance level that is typically used in neuroscience. So we thresholded them at significance of p less than 0.05, of course, corrected for multiple comparisons. And finally, we decided to also use a much sparser representation that is actually shown here below, that is known as the efficiency cost optimization, and instead imposes about no degree of 3 on average across the whole network. And that has been shown to have interesting properties in terms of highlighting the topology of the network, and it was proposed by Daviko Fallani and his colleagues in 2017. So as you can see, the threshold can vary quite dramatically. And of course, we're wondering which one of these is going to give us better results. We do not know which one of these corresponds to the actual connectivity in the brain. That is the problem. And then, final step, again, independent of all of the previous steps, is do we binarize our network? That is, once we have decided on a particular threshold at which we are sparsifying our network, and set everything that is below that threshold to 0, we still have the question of whether everything that is above that threshold should be left as is, preserving the weight, or instead should be turned into a binary network by putting everything that survives the threshold to unity and everything else to 0. And this is a schematic of the various pipelines that we have, and you can see that the result is 180 unique combinations of going from a brain to a network. Now let's have a look at some of the results. So here are the results for all the 7 data sets that we have. Here are each of the various measures that we have, each for all of these data sets. So each column represents a combination of measure and data set. And one thing that you can see is that this matrix of results is relatively sparse, as perhaps we should expect, given that we have unaccepted significant results here. However, you will also see that there are many cases in which some results are positive, that are in red, that is reflecting increases, and some results are negative, in blue, that is reflecting decreases in the same data. Now let's have a summary of that, because that matrix is not particularly viewer-friendly. So here on the left, you can see results that show significant increases for a particular metric, shown here, and for a particular data set, shown here. And as you will see, there are many significant increases, and everything is fine and good, until we look at the matrix of significant decreases. Because also in the matrix of significant decreases, we observe many of them for some of the same data sets and the same measures. And that's a problem. And here we are showing those inconsistencies. We're showing the extent of that problem. So every cell that is non-white here represents a case where some results, according to some particular pipeline, that measure in that data set is becoming increased, and according to a different pipeline, that measure is instead becoming decreased. That is, we have no idea what's going on whenever a cell here is not white. And the color bar here represents the extent of that inconsistency. And we measure that as how close are we to the worst possible scenario. Now, what is the worst possible scenario in terms of inconsistency? Presumably, we reason that will be when 50% of your pipelines give you a significant increase and 50% of your pipelines give you a significant decrease. That is the absolute worst scenario, because you have absolutely no idea what's going on there. And luckily, we do not ever get to that case, but we do get to 10% of that case, which is also not great. And the main other takeaway that you can get from here is that no single column and no single row is free of inconsistencies. That is, some measures seem to be better than others, and some data sets seem to have fewer inconsistencies than others, although if you look in the other two, you will see that those are also the ones where you don't see much in general. So it's not hard to have no inconsistencies if you're just finding no results. But that is the takeaway. That is, no measure and no data set is free from inconsistencies when it comes to network neuroscience. And one way that we tried to have a sense for what these pipelines are doing and how they are grouping together is by applying clustering. That is, we turned the set of various pipelines into, if you will, an adjacency matrix, and then we cluster them to see, are there sets of pipelines that go together that are consistent with each other but inconsistent with other sets of pipelines. So here a blue entry represents a case of inconsistency, and a red entry represents a case of consistency between the various pipelines across all data sets and all measures. So the good news here is that yes, there are sets of measures that cluster together and that are consistent with each other and not consistent with those in other clusters. That part's good. The less good part is that here are the, if you will, fingerprints of each of these clusters. And what you can see is that these are not easy to read. And that is bad news, because if we had seen a very clear-cut division between the different clusters, we would have a sense for what was going on there. But that is not what we see. Almost every possible choice is represented in almost every cluster. So we tried a different approach, that is, if you will, a simpler approach. So what we did is we kept, for each pipeline, we decided to change one or two of its steps and see across all the data sets and all the measures how many inconsistencies, that is, how many sign flips, do we observe as a result of that change. And you can see the results of that particular analysis here in these matrices. That is, how many, for instance, this cell represents how many inconsistencies do we see as a result of changing both the thresholding option and the binarization option. Now note that those within the diagonals can only be compared with each other and not with the rest, because in this case we're only changing one option. That is, only changing whether we use the Hilbert transform or only changing whether we binarize the network. And on the left, you will see the results that are averaged across the various data sets for all measures. And you will note that it doesn't look that bad. The worst case scenario is only about 4% of those are producing a change. However, this is, as I mentioned, averaged across all the various data sets, and there are some data sets where you find very little. So instead, this is a more representative result, we believe, and that is across all the various data sets and measures. What is the probability of observing at least one sign flip when changing a combination of our various steps? And in this case, you can see that it goes up to 0.25. That is, in a quarter, in the worst case scenario, in a quarter of our pipelines, in a quarter of our sets of data sets and measures, changing the network derivation steps will produce a sign flip. However, there is also some good news here, because we can see that some of the choices that one has to make seem to matter more than others. That is, it doesn't seem to matter very much whether you use the Hilbert transform or not. It doesn't seem to matter very much what node or edge definition you have. To some degree it does, but not as much as thresholding and binarization. So that is already some good news, because we know what to look at there. And you might wonder, well, that is all great, but do we have at least a sense of which pipelines we should go for? Because right now we just know what we should look for in general, what steps are the trickiest ones, but not where should we actually thread. And we do want to thread carefully. So we do think we have some recommendations. And the reason is that we noticed that there is, as perhaps one could have imagined, a trade-off between how liberal a pipeline is, that is, how many significant results does it find in the data, and how often it produces an inconsistency. Now, it should be clear that just finding many results does not mean that that is a good pipeline, because we do not know. We use the word liberality here because we do not know whether that pipeline is just finding a lot of false positives, or whether instead it's finding a lot of true positives. We do not know that, and that is part of the problem. However, we believe that given the kind of data sets that we have, we should have a reasonable amount of results. And it is not a good way to reduce inconsistencies to just adopt a pipeline that finds no results whatsoever. However, given that there is a trade-off between liberality and inconsistency, well, shown here are ranks. And so closer to the origin is better, that is a higher rank. So closer to first is better in terms of how many results the pipeline produces and how many inconsistencies the pipeline has. So this is, for instance, the worst possible case scenario in terms of inconsistencies, just to orient people on the plot. What we can see, though, is that there are some cases where the trade-off between liberality and inconsistency is better. That is, we can find pipelines that have a combined rank that is relatively lower compared to others. And if we do that, then we can find, for instance, the top 5 pipelines, according to that particular criterion of having relatively high liberality and relatively low inconsistency. And what we find is that all of those top 5 pipelines are using the Lausanne parcellation, for instance, and 4 of them are using binarization. And in terms of threshold, we find that either they're using the efficiency cost optimization, or they're using the structural density. Note that here for the Lausanne parcellation, which is what we use, structural density comes out to about 18% density, which is within the range of 10 to 25% that we use. So basically, what we can see is that about either a very, very sparse threshold from efficiency cost optimization, or about 20% seems to be the sweet spot for the Lausanne parcellation. And note also that these all use Pearson correlation. In some cases, it is the absolute value of the Pearson correlation. However, that is when efficiency cost optimization is used, which as a result of being extremely sparse is effectively identical to using the Pearson correlation, because there are no negative edges are going to be among the top about 1% in this case of all edges. So the Pearson correlation seems to be the winner in terms of edge definition. And so here are some recommendations that we can make based on this particular set of pipelines. That is, Lausanne parcellation seems to be the winner. Pearson correlation seems to be a good choice. And in terms of thresholding, one can choose between efficiency cost optimization and a structural based threshold of about 20% density in this particular parcellation. And most of the less inconsistent pipelines that we found here were using binarization of the edges. So to just sum up, the bad news is that there is a combinatorial explosion of steps. Every additional decision that you have to make will increase exponentially the number of potential networks that you will obtain as a result, and that these different steps and these different pipelines can produce diametrically opposite inferences in the very same dataset. The good news, if you will, is that some choices seem to matter less than others. The choices that seem to matter more are the ones that are at the end of your garden of working paths, such as binarization and thresholding. And using the balance between liberality and consistency as a guide to which pipelines to use can tell us something about what we should do, that is Lausanne parcellation, probably a structural density, and probably binarization. But these are, to some degree, tentative in the sense that they are only referring to the results that we actually have here. But, of course, there are many more options, because for each of the steps that we considered, there are further ways of getting, for instance, nodes out of a network. There are Glasser-Schaefer power parcellation. Some people use each voxel as its own node. There are different ways of using edges, such as partial correlation, wavelet coherence. So very many more options exist, and we've only sampled 180 of those. And of course, I should give thanks to very many people, because many people shared their data with us, many people contributed to the data analysis, and of course, I should also thank my funders, Cambridge, and everybody in my lab, and yourselves for your attention. Thank you. Any questions?